Yeah, good evening everyone. We'll begin the class now. I hope you're able to see my slides. So, the first question, it's just a general question. Like, we start with a historical introduction. The term chromatography was coined and four different options are given here. Since this is just a historical aspect, I'll go to the answer here directly. So, it was by this person called Michael Svet. So he's a Russian botanist. So he wanted to separate these plant pigments, for which he used a method. Later on, this method came up to become as this chromatography, uh, and you might have done this yourself, where in a tissue paper or in a filter paper, if you soak it in a particular solvent and add different dyes to it having different chemical nature, they would have different rates of migration. So this just tells you that based on the chemical nature, the compounds could be separated. uh, More introduction is given here in this Botanica page, you can refer to that. Now, getting on to the actual questions. The partial coefficient of stationary phase A and mobile phase B can be expressed as Kd equals dash, and four different options are given here. You can think for a couple of minutes and then maybe start with what partition coefficient is, and then we'll take it from there. So whoever knows the answer for this question, what partition coefficient is, you can start by unmuting. You can think for a couple of minutes and then unmute. Okay. Uh, does anyone want to try answering what partition coefficient is? It's right there in the name, partition and coefficient. Even if you don't know the answer, you can try uh, telling us what you know, even if you don't know the right answer. Okay, so uh, this is usually considered in the case of chromatography. So in chromatography, you have a solid support where the support is fixed, and you have a mobile phase which passes through, and your compound of interest is present in the mobile phase. Now, this partition coefficient tells you that if you have a particular combination of the stationary phase which doesn't move and this mobile phase which through which your particle flows, how much or what fraction of your molecules of the compound is interacting with the stationary phase and staying in the stationary phase, and how much of it is interacting with the mobile phase. This is what is addressed with the partition coefficient. Now, based on this, can anyone tell me whether we need to get concentration or charge, or a combination of both? Because the three different options given here, four different options given here, deals with different ratios, with either having fully concentration or having fully charge, or a combination of charge and concentration. So, can anyone think and justify what could be the correct answer here? What would prevent a molecule from uh, binding to a particular phase, and how can you estimate that? Think in these two aspects, you might be able to answer this question. Okay, so with respect to this, although charge is what determines how much of a particular molecule can bind to the stationary phase and how much of it can come to the mobile phase, we estimate this partition coefficient with how much it is bound. We will not estimate the charge, but rather we will estimate the concentration, which is why the right answer for this question is concentration of phase A by concentration of phase B. Right. So the explanation is given here. This partition coefficient represents how a compound distributes between two phases, like we discussed. And okay, what I didn't tell you is this KD value tells you where it is more likely to be present. If the KD is higher, it indicates that the numerator is higher, which means the compound has greater affinity for stationary phase. Whereas if KD is low, it means it will come in the mobile phase. Fine. A plot of elution volume versus absorbance is known as dash. Option A chromatogram, option B histogram, option C spectra, and option D volcano plot. Take a couple of minutes to think about this question, and then if you know the right answer, you can raise your hand. Okay, so this earlier we discussed this method called as chromatography. So the graph that we get from chromatography is the chromatogram, and it tells us how much of a compound has eluted at what time point, which you can correlate with what volume or elution volume. So a simple chromatogram looks like this. Uh, so here, what you need to notice is on the x-axis, you have time, whereas on the y-axis you have absorbance. So if you see a peak, it means something has come out of your column. Now, the thinner the peak, it means the lesser your compound has eluted. And if you see multiple peaks, there are multiple species that are coming out. And usually these peaks are uniformly distributed, like Gaussian peaks, Gaussian curves. And there is one concept that you need to be familiar here, which is your retention volume. So, depending on the nature of the compound, in your initial few minutes, you will not have anything that is eluted. And this volume is called as the dead volume. So, here, usually you will not have anything that gets eluted. But if there is something that is not at all binding to any of your uh, column components, that gets eluted here. Like for example, in this one technique called gel filtration chromatography, where we separate proteins of different molecular mass by passing it through columns, what we observe is if a column has a capacity to separate up to 200 kilodalton proteins, and if we add a protein that is 500 kilodalton, that will elute even before entering the column, because here the beads can only allow a particular size. Because this doesn't even enter the bead and doesn't even bind to it, it exits. And yeah, so coming back to the question, so depending on the solution volume and absorbance, the plot of the solution volume and absorbance is your chromatogram. The next question is, the formula for resolution of a chromatographic technique is dash, and 4 options are given here. So here, to start with, if anyone can tell me what resolution here means, that would be helpful. We can take it from there. So whoever knows what resolution in this context can raise your hand or unmute and speak. Okay, so we'll discuss what resolution is. Resolution tells us till what accuracy or what level of purity can you separate something. If you see here, this one peak is of very good resolution. It belongs to this compound called carb carbamazepine. So here you see it going to very high absorbance value, and there are no small peaks close by. Unlike this one, theophylline. In theophylline you see a small peak and then there's an increase. So this is of slightly lower resolution compared to this. This is called as resolution. And to calculate resolution, we use these different parameters. One is difference in retention times between the two adjacent peaks and the average peak width of the two peaks. So this tells us what is if the two peaks are merging or if they are not merging. So this numerator tells us if the peak center, how much the peak center is separated, and the denominator tells us the width of the peak. So if this resolution is lesser than 1, it is poor. If it's equal to 1, it means there is partial separation. If it is greater than 1.5, then there is baseline separation, which is complete resolution. Right. The next question is, ion exchange chromatography separates analytes based on their dash. Option A charge, option B size, option C hydrophobicity, and option D molecular weight. You can think about this question for a couple of minutes and then raise your hands if you know the answer. Okay, from the name itself, we can try telling this. So, ion exchange chromatography, it separates based on ions, cations or anions. And among these four different methods that are shown here, the ones that define an ion is charge. So, with respect to protein, if our sample analyte is a protein, then one thing that we could consider is the isoelectric point, Pi. So what we know is, depending on the isoelectric point and the pH of the buffer, the net charge of the protein can be determined. If the isoelectric point is lesser than 6 and the pH is greater than 6, it means that this protein can bind to negatively charged ions, which is why you have an anion exchanger. Whereas if the isoelectric point is lesser than 6 or lesser than 6 and your pH is lesser than your isoelectric point, then it can bind to positive ions. And we elute it with sodium chloride or potassium chloride, which has both positive and negative charge, K plus, Na plus and Cl minus. The next question is a relevant continue question from previous question. In cation exchange chromatography, the matrix has an affinity for molecules that are dash. So earlier I defined what cation exchange chromatography is and what anion exchange chromatography is. So now you will have to decide in cation exchange chromatography the matrix has affinity for what kind of molecules. That is, at that particular pH, are the molecules positively charged or negatively charged? Okay, like I told earlier, you have an isoelectric point. If your pH is above the isoelectric point, then your protein will bind to anion exchange beads. Whereas if your pH is lesser than the isoelectric point, then it will bind to cation exchanges. Here they so the kind of charge that you have also depends on this only. If at Pi equals pH, you have zero net charge. But as your pH goes lesser than your Pi, because you have more of hydrogen at H plus, which is protons, all your carboxyl groups in your amino acids will be protonated. Whereas your amine groups will get, instead of NH2, NH3 plus. So it will be positively charged. That is why it can bind to cation exchangers. And that is what answers this question. So in cation exchange chromatography, since Pi is greater than your pH, you have positively charged species which binds to your matrix. That is shown here. So in your cation exchanger, your stationary phase is negative, whereas your particle or your molecule of interest is positive. The next question is, in hydrophobic interaction chromatography, dash are immobilized on the matrix. Positively charged groups, negatively charged groups, hydrophilic groups and hydrophobic groups. You can think about this question for a couple of minutes. It's right there, but if you want to answer this, you need to also justify what hydrophobic interaction chromatography is. We have a detailed explanation that comes up later. You can think about this question for a couple of minutes. Okay, from the name as it suggests, in hydrophobic interaction chromatography, the hydrophobic groups are your stationary phase. Positively charged groups and negatively charged groups is what we saw earlier in ion exchange chromatography. Hydrophilic groups could be used in affinity chromatography. Now, on a related note, dash helps in binding of protein to matrix in hydrophobic interaction chromatography. So now we are trying to understand under what environmental conditions will a protein bind to the beads of hydrophobic interaction chromatography. Or to simply understand this, you can think under what environmental conditions will a protein's hydrophobic groups be exposed. Because, like you know, that like attracts like, hydrophobic groups will attract only hydrophobic amino acids. So, how you need to understand this question is under what conditions will hydrophobic groups within a protein expose themselves. Okay. When we talk about proteins, what we need to also remember is what other kind of molecules will maintain the structure of proteins. So, usually we have salts. Salts like NaCl and KCl, potassium chloride or sodium chloride. What this does is the salts are broken down into individual components in Na plus, K plus, and Cl minus, and it stabilizes by interacting with the water molecules that in itself interact with your protein. So, in hydrophobic interaction chromatography, what we do is we add extremely high salt. So now, when we add extremely high salt, yeah. So when we add extremely high salt, uh, your hydrophilic group is more solvent exposed here, and it is stable. And your hydrophobic group will look for if there are any other hydrophobic regions where it can bind to. And because your matrix has this hydrophobic ligand, it will bind to that. So this image shows that in the presence of high salt, more of your hydrophobic portion gets exposed and it interacts with your ligand. But in low salt conditions, you don't have that case. So this is the principles for hydrophobic interaction chromatography. So yeah, like I mentioned, it's given that salt promotes hydrophobic interactions. In high salt concentrations, hydrophobic interactions between protein and the hydrophobic matrix is enhanced. And they also talk about this effect called salting out, which we'll come in a few minutes. So here they say that the high salt concentration, the solubility of hydrophobic regions in the aqueous phase is reduced. So now they have to only interact with your hydrophobic ligands, like we discussed. And one more thing is they say that elution happens in hydrophobic interaction chromatography by decreasing the salt concentration. Right. The next question is 0.05% of sodium azide is used in column chromatography as it helps to dash. Option A, prevent microbial growth while storing. Option B, remove non-specifically bound proteins. Option C, for washing the column after elution. And option D, to equilibrate the column. You can think about this question for a couple of minutes and then we'll discuss what sodium azide is, what it could do to, what we can achieve by adding it to our buffers. Okay, so sodium acid can block this particular part of your transport chain within your mitochondrial membrane. So it binds to your cytochrome C, and it because this is needed for electron transport, and because electron transport is needed for cells to survive, sodium azide will prevent microbial growth, and we add very less quantity of sodium azide. And precaution is usually taken because sodium azide can also kill any cell that has these transporters, which means even human cells. Right. The next question is the partition coefficient Kd depends on dash. Only physical properties of the molecule, only chemical properties of the molecule, both physical and chemical properties of the molecule. It is independent of the physical and the chemical properties of the molecule. So in the very first question, we discussed what partition coefficient is. We'll come to that, but if someone knows something about this partition coefficient based on what we discussed, you can try answering this question. You can take a couple of minutes to think about it, and then we'll discuss. Okay, so first we'll discuss what partition coefficient is. It tells us whether a molecule can bind to your stationary phase better or your mobile phase. So for that we analyze how much of the compound is finally present in the stationary phase and how much of it is present in the mobile phase. Now this chemical interaction depends on the charge of the molecule. So this charge depends on the functional group, the pH, and anything other factors that are related to the charges, which means whether it can form a hydrogen bond or not, and likewise. But the position of these chemical interaction depends on the size and shape. So both the physical properties and the chemical properties is what determines the partition coefficient. The next question is, the increase in height of distribution plane will result in which of the following results? Increase in resolution of the column, decrease in resolution of the column. Resolution depends on the height of the column and thus no changes observed. Increase in number of distribution planes. You can take a couple of minutes to think about this question and maybe start with what distribution plane is, what is the theoretical plate, and then we'll get back to the question here. Okay, so here what we are trying to tell is, when we have a column, we have beads, and the beads are arranged in a particular fashion. Although these are in the form of liquid suspension, they are packed well. What we are trying to see is if we can quantify this and physically estimate these as plates. That is a theoretical plate here. So the number of theoretical plate depends on both your column length and the height equivalent to a theoretical plate. And your resolution will is directly proportional to the root of the number of theoretical uh, plates here. That is called as the distribution plane. So the higher the number of theoretical plates, the better your resolution is. You can assume this in a simple fashion. You have a protein of interest that is passing through a very long column. Now, because its distance is high, the number of theoretical plates are also high. And because the number of theoretical plates are high, it has multiple rounds of selection. That is one way you can think of this. Or you can say that it goes through multiple beads. So it is slow, and because these proteins are all of the same size of that species, all these proteins of the same species go through the same path multiple times, and then they elute. So it is of higher resolution. That is what is presented here. So since resolution is directly proportional to root of n, when h increases, n would decrease. That is, your efficiency decrease. And if your efficiency decrease, then your resolution would decrease. Right. The next question is, elution of bound protein from HIC can be achieved by option A by changing pH, option B by decreasing salt concentration, option C by increasing salt concentration, option D with guanidine hydrochloride. We can take a couple of minutes to think about this question. We earlier discussed the same concept. What happens? How do you make HIC work? So based on that question, you can think an answer for this question. Okay, so earlier we saw how at higher concentrations of your salt, your hydrophobic region is getting exposed. Now, to reduce the exposure, we remove the salt, and when we remove the salt, your protein folds in different ways. And one other way is water gets ordered, and your hydrophobic regions start getting buried. And once your hydrophobic regions are buried, they no longer interact. So that is how you do the elution of your protein in hydrophobic interaction chromatography. The next question is, chromatographic technique used to separate analytes based on their size is dash. Something that we discussed earlier. So I was telling you about how these proteins go into beads and then they get separated. We also discussed this when we talked about dead volume. So this is gel filtration chromatography, also called as size exclusion chromatography. So you have these beads. Extremely large proteins do not even go into beads. This is your dead volume. Whereas smaller proteins take a particular path because they have to go inside the beads and then come out. Although it is shown like it is just stuck here, it actually has a path within the bead. It goes in and then comes out. And depending on the path each protein takes, it will elute at a particular volume, which is the elution volume. Finally, the last question is, salting in is the process by which dash a high amount of salt increases the solubility of protein, high amount of salt inducing protein precipitation, low amount of salt exposing a hydrophobic group of proteins, low amount of salt increases the solubility of the protein. So to answer this, we need to understand what happens with different concentrations of salt and how the salt uh, changes the physical properties. So what you need to know is there is something called as hydration shell. So a protein will be covered by water molecule. Now, in the presence of salt, these water molecules could be replaced and your salt can bind there. So your protein will separate. But in extremely higher concentration of salt, it is these proteins that would interact, whereas your salt would interact only with the outer waters. This is salting out. So it just tells us whether you can precipitate proteins or not. So actually in labs we can purify this protein using this method as well. A very good salt that we use is ammonium sulfate because of the nature of the salt. There is a series that talks about the nature of salts and the nature of the ion in the salt, and both ammonium and sulfate followed by potassium is a good combination. So if you add that, that would actually eliminate or precipitate your proteins. That is the salting out method. Whereas salting in is the ability to solubilize your protein. Right. Yeah. So with this, we come to an end of set of today's questions. I will be on the call. If you have any questions, you can check with me.